Phenolic compounds are the most abundant secondary metabolites in plants. It possesses a common chemical structure comprising an aromatic ring with one or more hydroxyl substituents. The main groups of phenolic compound includes phenolic acids. Phenolic acid possesses much higher in vitro antioxidant activity than well-known antioxidant vitamin. Some examples of phenolic acids are rosmarinic acid, gallic acid, ferulic acid and many more. Based on multiple studies, it is shown that the biosynthesis and accumulation of secondary metabolites in plants are affected by environmental factors such as water, light, temperature, soil properties and chemical stress. Plant in vitro cultures offer many advantages over traditional plant cultivation methods such as fast growth, small plant tissue requirement, free of disease and virus, and it can be done throughout any season and time. For large-scale or mass propagation of plants, we can use plant bioreactor. Plant bioreactor refers to the use of transgenic plants to make unlimited quantities or commercially important substances. Plant bioreactor offers the possibility of monitoring and adjusting culture conditions required at different stages. The performance of bioreactor to support plant growth depends on several parameters such as pH, temperature, aeration and many more. This is also to ensure we are able to obtain high yield of desired secondary metabolites for pharmaceutical industry use. Dracocephalum forestei is a Tibetan medical plant that is native to the mountain regions of Yunnan province, China. In traditional Chinese medicine, aerial parts of D. forestei are used as diuretic, astringent, and antipyretic agents. Dracocephalum species plants are a particularly good source of phenolic compounds such as the caffeic acid derivatives like rosmarinic acid. The culture type of Dracocephalum species used in study is hairy root culture grown in woody plant liquid medium and in darkness. Regenerated shoots were cultivated on Mazagar 0.7% medium with the addition of 0.5 mg per liter BPA and 0.2 mg per liter IAA. The transformed shoot of D. forestei is cultured on a large scale in three types of bioreactors: plant form, Rita and nutrient sprinkle bioreactor, NSB, for test. The ideal bioreactor conditions are cultures in bioreactors were grown in mass medium containing saccharose and growth regulators. The shoots in all types of bioreactors were cultured for 3 weeks at 26 degrees Celsius. The expected outcome is that biomass of shoots increased significantly 40 times bigger after 3 weeks of growth in the nutrient sprinkle bioreactor, with significant amount of phenolic compounds, as well as rosmarinic acid. The second plant is Salvia viridiselle, also commonly known as red-topped sage, painted sage, annual clary and Joseph's sage, is an annual or biennial member of the Lamiaceae family. For centuries, it has been used as a remedy for infectious diseases and inflammatory disorders, especially affecting the eyes and oral cavity. Viridis is rich in biologically active chemical compounds. The aerial parts are mainly rich in polyphenolic derivatives such as phenolic acids, phenylethanoids and flavonoids. The culture type used for the plant species shoot culture, grown on Mars solid 0.7% medium with IAA and M-top. The shoots were cultivated in a bioreactor with plant form temporary immersion system in 2 different immersion frequency. Here we can see that there is significant growth and development of the plant species after a certain period of time. The most efficient production of bioactive compounds is achieved in shoots that were grown for 3 weeks in a batch system and shoots that were immersed every 80 minutes. This can be observed through the significant number of the total phenolic acid, phenylethanoid and phenol contents after 4 weeks of cultivation. Different plant species require different cultivation technique and approach. Approaches can be determining suitable growth parameters to support plant in vitro performances. This can be adjustment of immersion time and it depends on the study objectives. For example, if a study focuses on biomass yield or if the study aimed to yield high number of plant bioactive compound. Another approach is choosing a suitable bioreactor type and system for the plant tissue cultivation.